Hello, we're going to explain the ELMER algorithm in this video. So ELMER is an R bioconductor tool that's freely available and was firstly described by Lin D, Yao L, and Berman in 2015 in this article published in the Genome Biology journal. What ELMER does, it infers gene regulatory networks from cancer samples using both gene expression and DNA methylation. So briefly, we are going inside a cancer cell. We're looking for the molecular profile of that cell, and we're comparing it to a normal cell, and we are trying to understand why that cell is a cancer cell. So what's happening inside that cell that makes it different from the normal ones? So ELMER has mainly 3 steps, and the first one is to identify the methylated enhancers. So DNA methylation is the addition of a methyl group over the DNA sequence, so it doesn't change the DNA sequence, but with that addition of the methyl group, you can change the activity of the DNA segment. So you can imagine like the addition of a methyl group like a physical block, and then by blocking that region, the region is not like functional anymore. So imagine now that you remove it, that methyl group, and now the region is open and it can be active. So what happens is a master regulator transcription factor is able to bind to that region and then regulate the expression of a distal gene. So the second step of ELMER is to identify which is this target gene. So from the hypomethylated enhancers, it looks for the 10 upstream and 10 downstream genes, and it tries to see which of those are upregulated. And the third step is to identify which is this master regulator transcription factor bind to that enhancer and then regulate our distal gene. So this is the network ELMER tries to reconstruct. And ELMER has 2 ways to run it, the algorithm, the unsupervised mode and the supervised mode. What changes from one and the other is the information we have about the samples. So if you are comparing 2 groups of samples and you know they are homogeneous between them, among them, like you're comparing control cells versus treated cells, or a certain molecular subtype of cancer versus another molecular subtype of cancer, then you should use the supervised mode, which is going to use all samples in the analysis from one group versus all samples from the other group. On the other hand, we have the unsupervised mode. So imagine we have a group that's heterogeneous. So we say cancer versus normal. So imagine that cancer, we have different molecular subtypes inside it. And their DNA methylation profiles might be different, their gene expression might be different. Then if you take the mean, like certain regions might be only hypomethylated for one of those molecular subtypes. So if you have that case, you should use unsupervised. It's going to work on this distribution of those samples, which I'm going to explain a little bit later. But if you don't have that information that their samples are homogeneous, if you know that, you should use unsupervised mode. So the first step is to identify those different methylated distal probes. So what we call distal probes in the genome. So for ELMER, we're using 2 arrays, 450K and an epic array. And then we get the probes that are close to the TSS. So everything that's 2 kb away from the TSS, transcription starting site, it's removed. So we only keep the distal ones, that's what we define as distal probes. And then we're going to compare those probes and see which ones are hypomethylated. So normally those probes that are close to the TSS, they are normally in promoter regions, while the ones that are distal are normally in enhancer regions. So right now we have our distal probes, and for each probe, now we're going to compare the mean of group A versus the mean of group B using a t-test, and look which group has a mean methylation level higher than the other one. So this is the example for the unsupervised mode. So imagine we are comparing probe 1, okay? So you get the DNA methylation level of all normal samples and the methylation level of all the tumor samples for that region, and it's sorted, and we use the lower quantile of both groups. So this is, in a way, we consider we have different molecular subtypes. If we are using the supervised mode, we're going to use 100% of the samples and take the mean and just compare that. Okay, so our new hypothesis is simply if the mean of the tumor is higher than the mean of the normal. And we use a 30% difference of DNA methylation levels with FDR of 0.01. Those are the default values. So now we have our hypomethylated regions in cancers cells compared to the normal ones. And we go to the second step, which is the target gene. So we take a look at the 10 upstream, 10 downstream genes, as I said previously. And then for each one, we look for this anticorrelation. So are my hypomethylated or unmethylated samples with a higher expression of that gene compared to the methylated group, so the ones that have higher methylation, are they have lower expression? So we look for this anticorrelation. Hypomethylated, higher expression, and hypermethylated and lower expression. So we're going to have this correlation for the 10 upstream genes, and then we select the ones that are significant. We use FDR of 10 to minus 3 as default, but the correction way we do it is by a permutation approach. Simply, we change the probe for the gene that we found that was significant, and then we see how many probes would be having that anticorrelation pattern. So what we expect that if it's real, our probe and gene would be right in the beginning, so it's very significant for that probe and gene compared to all the other random 1,000 permutations, 10,000 permutations. Just briefly explaining, some people might ask why 10 upstream genes and downstream genes and not like 5, or why you don't make a window. So the genome is heterogeneous, and we have desert regions, which can be like 1 mega base pair. We don't have like any coding regions there, but they are regulatory regions. And imagine we are using a fixed window there, and we have the problem like we have desert regions and have gene-rich regions, right? So if you make a window around that, like a big window, you are going to get a lot of genes in the gene-rich regions and very few in the desert gene regions. And then there might be a bias that you can get like false positives in the gene-rich regions. So that's why we select 10 upstream and downstream genes instead of making a window, because the window width could actually change the behavior of the algorithm. And why 10 upstream and downstream genes? We actually compared to CHIAPET data, and that showed to be the best ratio between false positives and false negatives. Okay, so we have our hypomethylated regions now, which is the master regulator binding there. So we started by doing a motif enrichment analysis of the hypomethylated regions. We simply do a Fisher test comparing the hypomethylated probe versus the background, which would be the distal probes. And then we're going to have, okay, for all those regions, those are the most enriched motifs. So in this example, for example, you can see that the Fox families are very enriched. And that's actually expected, like if we have like Fox A1, Fox A2 as the top one, you also have Fox A3 and Fox A1, because if you take a look on their motif signature, they are very similar. They are within the same subfamily. They have like T, G, T, T. And then if we find Fox A1, we also find Fox A2 and Fox A3. And then that might just ask yourself, okay, we have a signature for Fox A1, Fox A2, and Fox A3. but which of those three, for example, would be the mass regulator? Could we infer which one is the most important one? And the way we do that is we get all those regions that have those signatures for FOXA1, FOXA2, and FOXA3. Okay, we get the average demethylation of those groups. So we get all regions, get the mean of them, and then we compare the expression of all transcription factor, human transcription factor, versus the mean methylation. So here's an example. We got the three transcription factors, FOXA1, FOXA2, and FOXA3. And you can see that the FOXA1 is the one that has this anticorrelation. So the samples that are hypermethylated, they have a lower expression of the FOXA1, while the ones that are hypo, they have a higher expression. So imagine, like, I lost that methyl group, the region is open, and the transcription factor is more expressed. So it's more likely that that's the mass regulator, because you increase its expression, so there will be a higher chance that that's the one that's binding to that FOX region. So we do that anticorrelation for all 1600 human transcription factors, and we rank them by anticorrelation. And this is visualized by this plot. We simply have the ones here on the top are the most anticorrelated ones, and from those we select the ones that are belonging to the same family. So you can see that we have, like, as the top ones for that region, like FOXA1, GATA3, ESR1, which are like cofactors, and the other FOX samples are like here at the bottom. So what we do is we rank them. We got the 5% of them and say, okay, which are the ones that belong to the same family? So imagine we have a FOX motif and say, okay, which within the 5% has this signature? Okay, it's the FOXA1. And since it has the same signature from the region, we expect that that's the one that's binding there. And so that's the explanation from ELMER. So the workshop we're going to run after, it's going to cover all those steps. There are more explanations about the algorithm in this paper from Lin D, Yao, and Berman. We also published the ELMER version 2 in Bioinformatics. And if you have any doubt, we can go there and take a look.